This is preparation for lecture 25. We're going to talk about somites for the most part, both in chick and mouse. They are quite similar in the two organisms. We don't have the timed appearance of somites every 90 minutes approximately. In frog or zebrafish, they develop kind of simultaneously as the embryo develops. So this topic is specific to the chick and the mouse. So this is a mouse embryo kind of unfolded, because of course you remember that the embryo, hopefully you remember, is a sort of a cup shape. And so the main thing to think about is that the node, which is kind of in this region here, remember that in the mouse embryo, cells are moving towards the anterior through the node, and whereas in the chick, Hansen's node sort of starts at the most anterior part of the primitive streak, but then moves back towards the posterior again. So those movements are different, but what's happening is very similar. So just prior to the node, either the node moving or the cells that are going to move through, there is this region which is called the stem zone. And in this region, there are neuromesodermal cells. So what this means is there's a mixture of both mesoderm and neural ectoderm in this region. And they are self-renewing. So, and as the cells then move out of the stem zone, they begin to be specified and then determined and so on. And you will get then neural tube cells, pre-neural tube cells that are present sort of closer to the midline, and then the pre-somatic mesoderm, which is more towards the outer area. So the stem cell region has a mixture of the two types of cells, but then they differentiate one way or the other, and they wind up in different locations depending on their position. And I think that might be everything that I was going to mention. Oh, just that this is sometimes referred to as paraxial mesoderm. Para means beside, and so this means that it's beside the axis. The axis is the sort of the midline through the body, and the notochord, you can't see it. It's below this midline, and so the notochord is referred to as the axial mesoderm, and the somites are paraxial. Okay, and I think that's pretty much everything. Now, there's a lot of work that's been done in the chick embryo, because it turns out that chick and quail are closely related enough and similar enough that you can make chimeric embryos. So embryos where you've transplanted some tissue from a chick embryo into a developing quail embryo into a developing chick embryo. And you can, the reason why this is so useful is that the cells look different. And so when you're looking at the embryonic tissue, the ones with the darker nuclei and bigger nuclei, really, those are the quail cells, and then the chick cells are the ones with the paler nuclei, so that you can, it's almost like doing a cell marking experiment, except that this is a natural marker, so it doesn't sort of dilute away as the cells divide. And there's also other genetic manipulations that are possible. We aren't really going to talk about them too much in class, but there's other ways that you can modify the embryo by introducing different molecules that disrupt signaling or something like that, and then you see what the effect is on the development. So we talked, I think it was in the last lecture, about being able to transplant the posterior marginal zone and have it induce a new primitive streak, a new gastrulation, as long as the transplanted zone was in a young embryo, where the primitive streak really hadn't gotten going yet, because there is this sort of inhibition that takes place. The Henson's node is not like that. You can transplant a quail Henson's node into multiple locations in a chick embryo with the Henson's node already formed. And you can get the induction of additional body axes in this embryo. So this is the regular one with the head folds, and then here's the beginnings of, I think, somites, if I'm correct on that. And then here are the transplanted ones, and they are starting to form similar structures. So they're a little bit behind. The Henson's node, this embryo was a little further along than the donor. And was there anything else? Oh yeah, if you choose, this is an important one too, sorry, I forgot about that. If you choose a later Henson's node, so in this quail embryo, this one is new, it's just starting, right? It's an early one. But this one, the node has retracted, it's about halfway or so down the embryo. And so it will form a new body axis, just like the original Henson's nodes, except that it's lacking the head tissue. So the age of the transplanted node makes a difference. A newer one can form the whole entire body, but as development proceeds, the gene expression that's going on in those node cells changes so that it can only produce a subset of the structures. And so this is just to point out that because you can do this transplantation and get a second body axis formed, this is very much like the Speman's organizer from the frog, but functioning in the chick, right? And there are some similarities. So the transplantation of the node gives the secondary axis, so you have these two little embryos forming. But at a later stage, you can't get the head structure, so you get more posterior. And that's just like what we talked about with Speman's organizer. So we're spending, I think most of our time is going to be spent on the chick embryo, but almost everything that that happens in the chick embryo is very similar or the same as in the mouse embryo. And so somites are transient structures. So they're blocks of mesoderm, but then by the time some of the later ones are forming, some of the earlier ones are starting to change shape a little bit because they start getting information about which part of the somite is going to form the dermis, which is right below the epidermis, what's going to be out on the lateral part and form skeletal muscle, and then what part is sort of more medial, so close to the midline, and is going to produce spinal cord vertebrae, so the bones of the spinal cord. And so each somite gets different signals that tell the different parts of the somite what they're going to form, and then the block kind of comes apart and starts moving around to the correct location. So that's why we talk about those as transient structures. Approximately 50 somites form. It's in different species it will be slightly different numbers depending on the length of the tail, for example, things like that. And it starts from anterior and moves to posterior, and a new pair form in the 90-minute cycles. They form to either side of the notochord after Henson's node has gone by. Okay. And so the first 5 that so the head doesn't, for the most part, have somites. There's sort of the head mesoderm, and that's going to form the head. But the first 5 somites actually just contribute to building kind of the posterior of the skull. So that's the part that they have. And then from then on, you have the neck, the thoracic, the lumbar, and the sacral somites, and they correspond to the particular vertebrae they're going to form. But of course, they do form these other structures in those places as well. And it's very interesting that I have this repeated twice. I guess I really want you to remember about this every 90 minutes. In humans, though, it's a much slower process, so it takes 6 to 8 hours in the human embryo for somite formation to take place. And this is kind of a schematic. So this is Hensen's node, and this is the sort of closing up of the primitive streak. Although something to keep in mind is that while this process is taking place, the embryo is actually growing and lengthening. So, you know, as Hensen's node moves, the embryo also grows a little bit longer. So you're seeing a lengthening of the whole embryo plus the movement of Hensen's node in there. And so this is the mesoderm that's anterior to the somites, which is going to form the head. The first 5, as I said, they contribute to the posterior skull, and then somite 6 onward is going to be the vertebrae, the neck vertebrae, the cervical vertebrae, the lumbar, which is the kind of your thorax, the thoracic, and then the lumbar, which is your lower back, below where the ribs are, and then the sacral. Okay. And it turns out what's really interesting is this is called the pre-somitic mesoderm, and it's defined as the region between the most recently formed somite and the position of the Hensen's node. And as this process proceeds, Hensen's node moves, a new somite pair is formed, and but the region of the pre-somitic mesoderm remains constant. And I have a very ugly diagram which might help. So this is our embryo, and suppose this is where the node is. It's not really to scale, but this is your region of pre-somitic mesoderm, and here's your somites. And say just the first pair has formed, and this is the region. Now this mesoderm, quote unquote, knows its anterior-posterior position, and we'll talk about how that happens towards the end of the lecture. So there's information in here already that you're going to form somites, and this is the position of your somite. As additional somites form, the Hensen's node has moved further down, and I should have really, like the embryo would have grown a little bit too, so the embryo lengthens at the same time. There's better pictures coming up, but I sometimes find these sketchy ones are helpful just for explaining. And so there's 2 more somites. The node has moved along about the same length as those 2 somites, and so this it actually doesn't look the same size, but I copy pasted it so it should be. But in any case, just assume that this is the same size. In our pre-somitic mesoderm, it's the same length, but it's just moved further towards the posterior. And the size of it is approximately the same size as about 6 pairs of somites. So I clearly haven't done it to scale. I've made it about the size of 3 pairs of somites. Okay, so somites, when they develop, they have an anterior-posterior specification. So they are beginning to express the right genes, or they will be expressing the right genes to form the correct structure. So the vertebrae in your neck versus your tailbone are very different in shape and structure and so on. And the somite that forms, before it even forms, has the information that it's going to form part of the neck, part of the thorax, part of the lumbar region, and so on. So it's been given those kinds of instructions as the node is moving through. And there's a couple of different experiments that can be done, and we'll talk about these. And we'll talk about both transplant experiments that show the fact that the somites have this anterior-posterior information, and it can't be changed once they reach their new position, but also that the timing of their formation is kind of set as well, and it's sort of interesting. And so this is something to think about. So if you take a pre-somitic mesoderm in a later stage embryo and transplant into an earlier stage embryo, then when you see the somites forming, they actually produce the structures that they should have formed in their original position, rather than changing with new information at the new position. So they're forming the wrong structure. So you want to think to yourself whether those transplanted cells were specified or determined. That's one of the sort of underlying themes of the course. And I'll just point out, just because I'm not sure everybody knows this necessarily, this is in the human, but in the mouse, the names of the vertebrae are the same. The neck ones are called the cervical vertebrae, the thoracic vertebrae. They're important because they actually have ribs, the thoracic ones. Lumbar is the lower back, and then sacrum in humans is just a couple of fused vertebrae that make the tailbone. But of course, in the mouse, the sacral vertebrae form, all the structures in the tail. So there are more of those in some other species, of course. Okay, and so if we take from an older embryo the presomitic mesoderm, you've already got a bunch of somites that have formed here, and then you transplant it into a much earlier stage right behind the first pair of somites. Then what you will find, and notice this is only transplanted on one side, because that way you can tell if it makes a difference, because the somite on this side should be forming the correct structure, right? And so you get these funny sort of hybrid vertebrae, because on one side, the vertebrae that are formed have ribs on them, up in the neck where they're not supposed to be having ribs. But on the other side, the somites contribute to the formation of the other half of the vertebra, and they are normal neck vertebrae, right? So this just shows us that the mesoderm that's grafted into that younger embryo develops according to the original position rather than the new position. And there's also excuse me, a timing issue. And so if you take the presomitic mesoderm in an embryo at a particular stage, so you excise it out, but then you just turn it 180 degrees and flap it back down, right? So this is more anterior and this is more posterior, and when you transplant it, the anterior is now in the back and the posterior is in the front. And so what's going to happen, right? So what actually happens is that, so suppose these are the first 5 somites, so then this would have been the 6th somite. This one forms next, which is very interesting. And then this one forms after that, and then this one, and so on. So they form in the wrong order, and then once the grafted tissue is passed, then they get back to being formed in the correct order again. And so again, this tells us that there is specification on the timing and the identity of these somites that takes place before they form, right? So there's an internal kind of clock mechanism. Now the model that I'm going to show you for the development of the somites, some of it doesn't quite explain, or at least I would find it hard to reconcile this experiment and the timing stuff. But it's a very interesting thing, right? So that there's a lot of stuff going on in this region that that tells this mesoderm what it's going to form and when the somites are supposed to form. And the first signal that we're going to look at is FGF. There should be a nice figure in your textbook that just summarizes FGF signaling. It's one of the phosphorylation cascade type signals. Okay, so these are steps in chick embryo, and it turns out that there is an FGF and Wnt gradient. So both of these molecules are part of the gradient, and we talk mostly about the FGF side of it, but Wnt is there too. And so what happens is the node itself is a source, of FGF. And the FGF is a diffusible ligand. It's a short-ish range diffusible ligand, but there's also a degradation of it. And so so it's being produced here, and the cells nearby are receiving small amounts of the signal, and the signal is basically degenerating. There's less and less of it as you move towards the somites, and it gets broken down at some point, right? And so the further you are from the node, the less signal you have. So this is what this gradient is referring to here. Yeah, and so I'm just going to leave that, and then we'll go to the next one, because this is, so after the first pair of somites form, they are also a source of a retinoic acid gradient. Retinoic acid, some of you have heard about, because it seems to be, or has been in the past, one of these fancy cosmetics, retinoic acid, look young forever kind of stuff. But retinoic acid is an important signaling molecule. It's a precursor for vitamin A as well, so it's an important substance. And yeah, I'll leave that. Never mind. I don't want to start going on tangents and wasting time. So basically, the somites are a source of retinoic acid. And again, just like any diffusible signal, it's going to decrease as you move away. So the last pair of somites is the last place where you have high levels, and then the signal degrades as you move away. So there's less and less of it, because it can only diffuse so far, and there's less of it. And so essentially, the place where the next somite is going to form is defined by having a small amount of retinoic acid and having a small amount of FGF. And in that region, at this region, this is where the somites are going to form. So there's a specific threshold, right? And beneath that threshold is where you're going to get somite formation. And the other thing that retinoic acid actually does is it's an active sort of repressor for the expression of FGF and Wnt genes. So this prevents the presomitic domain from getting too large, right? And so you've got a signal from the somites that's keeping that amount of presomitic mesoderm limited. It's preventing FGF signaling from extending too far. And then you've got that region of overlap between these two, which is where your next threshold is going to form. And then after the next somites form, now the retinoic acid gradient pushes forward like this, and at the same time the node will have moved further on, and so the FGF gradient starts further back and it degrades further forward, and now this is our next region of overlap, and this is where the next somite forms. And then this all moves forward, this all moves backward, and it stays the same length and the same sort of size of overlap, but the whole thing just shifts as each new somite is formed. I hope that's clear. Sometimes things like this, you have to kind of go through them a couple of times to get the idea. And so you've got FGF and Wnt from the node side and retinoic acid from the somite side, and they interact and they basically form this FGF gradient, because the retinoic acid, when you have lots of it, there's no FGF, and then where there's less of it, you get less of the repression, and that leads to the gradient, right? And then, as I said, the somites form at this particular threshold is when the FGF drops below a certain region. That defines sort of the posterior boundary of the somite. It's actually good that I accidentally flipped back to this slide. Okay. Oh, that's weird. And there are also cyclical waves of gene expression, and they're activated from posterior to anterior. And each cycle determines the timing of when that somite forms. So we know where it's going to form based on the FGF, the low FGF signal, but the wave of gene expression takes about 90 minutes, and that tells us when that posterior boundary is going to form. And the gene expression, there's a lot of node, FGF, Wnt, and nodal, I think it is, signals. Notch, sorry, Notch. And there's a ton of downstream genes from these kinds of signals, and there's many of these genes that go through these cyclical waves of expression based on FGF, Wnt, and Notch signaling in the cell. So we're not going to look at like particular individual ones, although I posted a video done by a former grad student and TA for this course and for other courses that I've taught, and it does include at least one of the genes that is expressed in a cyclical manner. So here's the example. This shows two kind of full cycles. So here's our somite forming. This is the posterior margin, and that is at the lowest level, like below the threshold of FGF. So that's where our somite is going to form. Okay. And this is the finish of a particular wave of gene expression, and the most anterior point it reaches is coincident with that threshold. So it's the most anterior place the gene expression wave gets to, and the threshold of FGF below a certain level that defines that posterior boundary. Okay. And then there's so that forms, right? And then there's a new set of gene expression that starts at the node, and then it just waves up. So it's transmitted from cell to cell, and it moves towards the anterior, and then the furthest that it reaches is now coincident with the low level of the FGF gradient. And then the next somite forms. Now the gene expression continues up here afterwards, as the somite is forming. Okay. So that's one cycle, and then the next cycle. So again, notice how the node is moving down because the embryo is lengthening as this process takes place. So, and here's the node, and then here's our next wave of expression. And the dark is more of the gene expression and the light is less of it, so you can see how the large amount of it is moving towards the anterior, and it's temporary because as it's moving here, then that gene expression is turned off. So these cells signal, these cells signal, these cells signal, and so on, and it kind of moves towards the anterior. And then again, it reaches this boundary below the threshold for FGF expression that defines the boundary, but then as the somite is forming, then there'll be a little burst of gene expression in there again. And so this is just two cycles of somite formation. Now, it's thought that the identity that a particular somite takes on is related to the number of waves it experiences, because cells that are like up here that form the second pair of somites, they had 1-2 waves of this gene expression, but the next one had a third wave, and the next one had a fourth wave. And so it's thought that some of the information on where you are anterior versus posterior through this whole process is like, this one will have experienced 1, 2, 3, 4, 5. So this one will have had 7 waves of gene expression, and so it has a certain identity, right? Cells down here will have had 8 waves, and so on. And so that's part of how the the somites, when they form, have their anterior-posterior specification. Okay, I'm going to stop there and start a new one, and I'm going to try to be less chatty and more focused for the next one.